Thank you guys for coming today. Today's the first of a series of lectures, training sessions over the Access Grid. They're put together by the Molecular Cell Biology Core of the BRIN program. These sessions will be followed at a later date by hands-on training modules where we can bring you in and do training here at LSU and give you the training you need to bring back to your labs. Each of these sessions will be recorded for playback at a later time from the LBRIN website. And so we'll get started today with Pete Mottram from the PBS department at the School of Veterinary Medicine at LSU. And he's going to talk to us a little bit about the facilities in PBS and the PBS BioMMED Microscopy Facility and about a topic of interest in microscopy for him. Thanks, Pete.
Thanks, Mark. My name is Pete Mottram. I'm a coordinator of Pathobiological Science Department over at LSU School of Veterinary Medicine. I graduated with Allied Health degree at 92 and worked in intensive care in a hospital setting. And in 95, I graduated from Southeastern pre-professional zoology degree with chemistry minor. 98, I graduated from LSU-S in Shreveport, biochemistry, molecular biology degree. And I was able, at that time was where I met Javier Alvarez, would be the second presenter. He was running the microscopes over there at that time. I recognized my friend there. And then I went to work at Tulane for about 10 years and had the opportunity to work with microscopy, fluorescent microscopy there. And then now I'm working at LSU over here for the past year. So just made my first year at the PBS department. And in that year, they've expanded the microscopy basic fluorescent area of the center that we're calling it in the department. And we've have several different microscopes and different things that we're trying to do over there and expanding as we speak. So that's just a little background on who I am. So in the basic center for the fluorescence, we're not using any scanners or anything, just basic CCD cameras, digital charge couple device cameras. It kind of is like a two-dimensional grid that collects photons and assigns them to a certain spot on that grid, and it gives it to a digital code, and we're able to interpret that through the software as a light and convert that to fluorescent emission. So one of the microscopes, one of our newer microscopes that's kind of exciting is this Olympus MVX 10 Macroview. It's like a giant fluorescent dissecting stereo microscope, which is pretty cool. We've only been using it now for a couple of months, but a lot of the researchers have found it very instrumental in what they're trying to do, looking at fleas and different things, insects and things. So this thing has the white light source, halogen source, in addition to a UV light source, and with three colors: red fluorescent protein, green fluorescent protein, blue fluorescent protein. It has a mode where it can go from stereo to mono as far as the visual plane. So it has a 1x objective and a 2x objective, and it has a mechanical zoom. So we can achieve, I think, up to like 37 or 31 zoom factors. So it's really something. We can go from a very, very small, as an example, you can see the entire tissue there, and then down to a magnification of the fluorescent. And we could do that with the fluorescent dyes or genetically installed proteins, or also would just for a macroscope for the sake of dissecting and things like that. It runs with the Olympus software, the DP72 camera. It's pretty user-friendly. We have sign up. It gets a pretty good little bit of traffic. This particular microscope is beginning to gain popularity over here at LSU, and some of the groups are designing experiments around this particular piece of equipment that they can utilize it. Okay. So these are just, we have a lot of different scopes over there. This is a basic Olympus IX2 inverted microscope. It does a bright field or fluorescent. This particular scope we purchased with the idea of being able to get three-dimensional DIC type images through plastic flasks and petri dishes and whatnot. So Olympus has a function they call relief contrast, and that's designed to optimize the three-dimensional contrast through the plastic layers. So this would be kind of a representative image of the benefit of relief contrast over phase contrast. On the right, you see the nice image there with the relief contrast as opposed to what you could achieve with the general phase contrast. This particular microscope has phase contrast and relief contrast. And it's also easy to use. A lot of the researchers are going to the easier to handle microscopes, and the softwares can sometimes be cumbersome, and that particular Olympus system is very easy to use, and they're not having a whole lot of problems and hang-ups with the software and the cameras and things like that. So and this is our upright Leica DM4000B compound scope. We've configured it in this image. This picture shows configured with the condenser for bright field. We've turned this into our dark field station. It'll do dark field or fluorescent microscopy. We've installed a high resolution dark field condenser and the 100x objective, because one of the groups is actually trying to image spiroplasm, a very, very tiny organism, and trying to count them and things like that live. And the only way to do something like that is in the dark field. So it's all digitally controlled, except for the stage is not. It's a manual stage, and we're running several softwares on it right now. But the Image Pro software, Image Pro Plus version 5 is the one that we're using to analyze these things. This is just an example of the live cells in the dark field that one of the researchers is doing. It's able to you don't have to fix the cells. That's the big advantage here, and you can get a good look at the number and size and things like that. Okay, and then the other Leica system we have is the DM-IRB inverted scope. This is a much lesser resolution version of the dark field and bright field, also phase and fluorescence. We have a variety of objectives on that particular microscope, and it's a little bit older model. Everything's manual on it, and we're using the Q-Capture software as the main software for analyzing with this particular setup. It's good for it's inverted scope, and the condenser is mobile. We can move it up and down, and we put a lot of flasks and things. People use this one particularly for larger flasks and dishes and things. You could even probably put a small bottle in there if you liked. And just an example of a recent experiment a fellow was doing. We get a nice contrast and resolution there with that particular scope, and the different colors are real nicely done. So then those are the lower end scopes. And we have a setup for configuration for live cell microscopy over there. We've partitioned a room where we have the Zeiss Observer Z1 set up. It is equipped on a vibration-free table. It floats on a pressure of nitrogen gas. Try to minimize the amount of fluctuation and drift and things in the focal plane. And then it's set up with CO2 incubation. We can control the temperature, the humidity, and CO2. The air on the stage. It allows us to take a flask or a dish, petri dish or a plate with live cells, and we equilibrate them to the temperature in that in a special incubator compartment that sits on the stage, and then we can go ahead and focus in on whatever cell types we want to visualize over a time-lapse period of time. It's kind of interesting. Has a wide variety of filter cubes. We have 20 or so different combinations for excitation and emission. It lends itself to be functional in most applications. Every now and then we have to buy a new cube. It's a couple $100 if we happen to get a dye set that doesn't match what we have. It's not that big of a deal to get the right filter sets. There's a variety of objectives that go with this particular scope. We have long distance objectives that are designed to get through the plastic layer of a flask, or say, where a regular objective wouldn't have the working distance to peer through the thickness of a plastic dish or something, with special objectives for that. We also have a special module, the Apparatus. It's a very expensive add-on for this type of scope. It's basically a slider that slides into the side of the scope, and it goes into the light path, and it sets a grid in there. The grid itself, when the microscope, the computer focuses on the edges of the grid, and then the grid shifts just a fraction over, and it refocuses on the edges of the grid, and it shifts again and refocuses on the edges of the grid, and then it recombines all the focuses in the theory that the edge of the grid has a higher resolution than the center of the grid line. So it combines, it uses logarithms and things like deconvolution. It's similar to deconvolution, and recreates a slightly higher resolution image through this apotome. It's a little complex, but we have it working pretty well. It's functional. To the trained eye, you can see the difference. To the untrained eye, it doesn't look like that much of a difference. But we do have applications where the researchers are hoping to get a little higher resolution, slightly higher resolution, and we go to the apotome and try to achieve that for them. This particular scope is fully automated, or fully mechanical. So we have a motorized X, Y, Z, motorized everything. The objective turret's motorized, the filter turret, the aperture, everything's motorized. So we can control it completely from the computer set on in a separate desk. So we don't have any vibration there. And once we have an X, Y, Z motorized stage, it opens up for a lot of different functions. When we can start doing stacks of images and create three-dimensional images, and it's almost like it turns robotic. We tell it exactly what to do, and it'll collect images from different areas, and then it'll make stacks of images at different areas at different times, and it's very, very accurate. It goes back to exactly that spot because of the motorized XYZ stage. So these are some of the apparatuses that goes along with the live cell. This, you can see the 6-well on your left, the 6-well plate, and then beneath it, you can see two different petri dishes we can set inside of this lower one, you point or what, like this. I guess, I don't know if they can see me or not. This is an objective heater, and the idea is to keep the incubator, the plate, and everything at the same temperature as if you put a cold objective alongside a warm plate, it drags the heat in. They call it a heat sink, and it causes drifting and things. The cells will walk away sometimes. They feel the temperature difference, and they start to migrate if they can and whatever. That's the incubator lid, and you see the humidifier chamber, basically just in line, and just still water, and that's also heated so that the air.
You pick up the pen.
I can draw circles.
And then they'll see.
Okay.
As long as you put the pen back to get control.
Oh, all right. Okay. So we heat the water, and when I put it back, it goes away. You said? How do you reset it?
If you want to erase it, there's a little eraser.
Oh, right.
Right.
Yeah.
All
All right.
right.
Oh, never mind. But you can see, I think that's the only thing that I had to point at anyway. And there's a module. We control all of these devices with through the computer, and we could have it change and things like that. Monitor the status of the temperatures and things that are going on in this incubation. It's really, it works really good actually. It equilibrates. Within a minute, it reaches the temperature and the desired temperature, and it maintains pretty well. We haven't had a lot of problems with the incubator setup yet. Initially, I thought it might condensate and whatever on the top of the incubator glass, but it just doesn't. It's somehow it maintains visibility in that. This is just an example of one of our researchers is using equine herpes virus. It's a triple. He's got 3 different proteins genetically structured in the virus, and we've applied it to some Vero cells, and we're playing with it right now actually to optimize the capturing of this particular virus in different settings. And this is just an example of the 3-color virus, and then in the live cell, this is from 6 hours post-infection to 20 hours post-infection. It's just an accumulation of these proteins, and as they accumulate, you see the fluorescent increase and spread. It's pretty groovy. Yeah, that's what I spend my time doing these days. I like it actually. But it's not. Javier's going to have some really nice videos to show, so you can't show that one twice. Okay, but there's a live cell. So the Zeiss is a really sophisticated piece of equipment. It has a lot of potential. It does a lot of things. The software allows us. We have a physiology module, and we also have a mosaic module, along with the live cell modules and the apitome things. So the physiology is really interesting. We have a specialized light source that allows us to switch the excitation wavelengths within a nanosecond of each other. So we can go from green to red in nanoseconds, basically simultaneously, and capture the emissions on that and monitor different physiological activities that are taking place as they take place over monitoring different antigens or different reaction of quantities of calcium or whatever, pH changes or whatever it is that we want to look at in that case. And it reads out as a ratio over time. It's pretty interesting setup. We can assign different changes along the way. We can say at 8 hours we want to change, the parameters of this, capturing this data, whatever, and then change it again or whatever. There's a lot of functionality there. The mosaic, a mosaic function allows us to focus on the field that we'd like to see, the visual field, and then so we center on, say, 2 cells doing their thing, whatever it is, and then the mosaic will capture in a grid kind of fashion several pictures, several fields surrounding that. So I can center on the two cells, and it'll get the upper left hand, the upper right hand, the lower right hand, the lower left hand, and it'll keep going. I could spread out and have the thing robotically capture all of the surrounding areas in that, and we can catch that in 2-dimensional, and because of the motorized Z, we can capture on several different focal planes, which later can be combined into 3-dimensional images. How am I doing on time? Oh, I'm doing not too bad. Okay, so then we have the pathology group over at LSU was really interested in this Nanozoomer machine, so we bought an Olympus Nanozoomer from Hamamatsu. Olympus deals Hamamatsu in the USA. It's kind of really interesting thing. I wish I'd have had the Nanozoomer a long time when I was over at Tulane. If I had the Nanozoomer, it'd been a whole different data portfolio there. But what this thing does is it scans the whole entire slide. It'll mechanically, or you could manually tell it where the tissue is. It gets the whole slide from the label, the entire thing if you like. And so then what you can choose, looking through a microscope at a field of vision, that's 1 millimeter or 4 millimeters, in diameter, and then looking at the entire slice of tissue, two different things all together. So it comes in really handy. What the pathologists are doing with this particular is they're archiving all of their old H&E slides and things that they have stained, different DAB stains and different animals or whatever, disease states and they have or whatever, and it becomes a digital file. The entire slides, you can zoom in from, the whole slide to 40x and then some, and then go even digitally even lower than that. It's real handy with pathology as far as antigenicity and things like that. Maybe somewhat handy for maybe cell counting or determining the quantity of antigen or expression or something you may have in the tissue as compared to the whole tissue slice. It's the see in the forest, as the forest, rather than looking at the tree like we do a lot of times with microscopy. So this will go 20x or 40x. We can run 210 slides automatically. We put it in batch mode and just let it go, 200 and something slides, and it just picks them up. Robot slings them on over there to the light source, and the lens does it. The lens moves back and forth across the thing. The software stitches the images together, and then finally you have a file. It can range anywhere from a couple of gigabytes to a lot of gigabytes, depending on how much detail and if you set it for the 40x and whatnot, and you want a big slice, and you could do several. You could do Z-stack. It's got a motorized Z-plane, so you could do Z-stack with that also, and then the file types could even larger. The Z-stack is what happens is you set the focal plane on a focal. You focus on an image, and the Z-stack will move the Z. You can tell it exactly how much, say, half a micron. So I want to, call this my main focal plane. I want to move a quarter or half a micron up, another half a micron up. It takes another image, another half a micron up. It takes another focal plane. So eventually I got 6 microns with 48 focal plane images, and that's where the 3-dimensional comes out of. Okay. And this is just a picture of what the entire piece of tissue looks like. And there's this VisioFarm software. It's just one of these expensive software packages, $40, $50, $70,000, something like that. And it's for analyzing these super images, they call it. When you capture the whole slide, they call it super image. And then you go down from your super image, in this case, to to this would be a virtual area, and then these here, it's sampling virtual area here. These little boxes, you can tell it to randomly sample, things like that, and then you can take, like when you have a big, say you want to count the.
Because there's an eraser on the left.
Yeah, I'm pad with the eraser?
See, on the left, pick up the
Oh,
eraser,
this
and then you
thing
can
right
wipe
here.
it.
Okay. Oh, very nice. Okay.
Or put it back.
Yeah. Alrighty. So, if you try to count the number of, you go and quantitate the number of DAB, expression in that entire sample. A lot of times they find it convenient to take 10% of the entire sample and then in little box sections like that, and then it'll read each one accurately and then combine them later with the data set. You can define every little parameter of everything you want to do or thought you might want to do, and so the software is real powerful, real sophisticated, complex software, and it's really good stuff. And in different software packages that we're using, the Image Pro again, which has a variety of functionalities. The Capture Pro, which is easier to use version. The Axio Vision by Zeiss, which has its own Zeiss specific stuff to do. The Visio Farms, the Nanozoomer Hamamatsu product, high-end analyzation software stuff. And then this DP2 Olympus, which is really easy to use. Not too much analyzing going on there with Olympus. So these next couple slides are just examples of different ways that I've been able to use this fluorescent microscopy and other microscopy things over the years. In this particular slide is some stuff we did, some monkey brains over there at Tulane Primate Center. And we showed the inflammation areas of these brains, and it's a long story about what the experimental procedure was, but what this is sequential slides. So when when they go to cut the block, I request slides, because I want to stain them. So they give me, I have them number the slides. So from that same block, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. If they're together, numbers 7 and 8 basically came from the same. They shared a common slice there. So the tissue is not going to change very much from one slice to the other. Anyway, so that helps a lot when I'm trying to demonstrate the morphology that we could see with the H&E staining, and then we want to show antigenicity more specifically with the fluorescent. This being a section of the brain that has these blues happen to be B cells, and then the greens are T cells, and the reds are macrophages, and it's related specifically to that same area of brain tissue there. Then we use it to demonstrate cellular functions. In this particular experiment, we're showing in tumors, we were showing apoptosis. How these particular situation was inducing apoptosis inside a tumor cell, or whatever. So here you can see the apoptosis going on. The cells are blebbing. They've bursted in the DNA. What it's actually reading is the DNA. The DNA's coming through the cell membranes, gone, and it's starting to blob out like a bleb. And then here we have the CD3 seems to be trapped in between the two blebbing tumor cells, and that was real important for what we were trying to say about what was going on with these particular molecules. But you can see clearly the functional apoptotic stuff going on, and the relationship between the apoptotic cells and the CD3 T cells. And phenotyping, sometimes like this particular, this was a novel cell that had, nobody had actually shown it before, and we were able to demonstrate the phenotype of this cell, and it was real important for the guys that were trying to do that. And so we were able to show these particular cells, cell types by the antigenicity. And the important part was this FOXP3 is a transcription factor, and it was happened to be, so it's an intercellular product. The CD25 is an extracellular membrane product, and in the location they define it that the antibodies actually work nobody had ever seen these before, and we happened to get lucky and be able to show the FOXP3 in the right location. If I had shown it outside the cell, it would have been artifact, but because it's in the right location and it's in the right area and everything like that, it was decided that it was a real staining, and it was very important at the time for those guys that were trying to show that stuff. And again, the same group of experiments, but that was really important bunch. So they showed it in function. So these are the particular cell types interacting with CD8 cells. So what we're showing there with the fluorescent microscopy is the reality that these particular FOXP3 cells are in physical contact with the blue CD8 cells, and it caused inhibition there, immunological inhibition, which was extremely important to those guys that were studying that stuff at the time. Okay, and then another function for this type of microscopy is quantitation. A lot of guys, when we get to we've defined the cells, the stains work, we're looking at tissue, you really want to know how many of these cells are there. You know, it's a big question, and it's hard to tell when you look when you're looking at that 3 millimeter segment of some big tissue slice. So, so what in the past we've done is manually count the cells, and it's very tedious and labor intensive. A lot of the researchers are shy away from manual count. You know, I happen to have done a whole lot of manual count, and it's not that tough for me to do because I got to develop the system. You know, I sequentially go through like a robot and go from one field to the next and shift down and count each phenotype and lock it in and go back later and quantitate it. It can be very, very powerful tool. You know, it's hard to validate it because in microscopy you're looking at, you know, that one little tiny 3 millimeter two-dimensional section and try to infer that that has anything to do with the entire tissue organ or whatever. But it's what we do with microscopy. The nanozoomer helps out because you can actually see the entire thing in 2 dimensions or 3 dimensions if you like. And then I think it has a more realistic picture of what's going on, a little less subjective, which this microscope is all about. So this shows, in this particular example, they shipped me 150 tumor samples from all over the country. I had this dream that I woke up and I was unwrapping bubblegum wrappers and there were tumors in it. And I was like, man, this is a nightmare. And then like next week, my boss is like, yeah, we're going to start shipping you tumors from all over the country. And sure enough, there it was. Here come bubblegum wrappers coming in left and right. We logged them all in, sliced them up, stained them all up for this particular cell type that was so important to these guys, and quantitated 100 different tissue samples, 100 different tumor blocks there. And it was a lot of work. They were real happy. This is another type of quantitation. Or this is a qualitative fluorescence with co-localization. Here you'll see, and I'm going to show you briefly, but I'm sure Dr. Alvarez is going to have a whole video series to go with this. But just real basically, you know, you can see clearly, this particular antigen is expressed across the tissue, you know, and then the red is a macrophages, you know, and this green is this experimental novel molecule at the time. And on this particular slide, you can see that the macrophages also express. When you put the green and the red together, you get a yellow color like that. And you can see most of the macrophages are expressing this green because they express it as a yellow, which is the green plus the red. In this particular group, another series of Penn State sent me 72 tumors in bubblegum wrappers, and we sliced them up and stained them up real nicely and tried to compare them, which is a real task. And so I was able to quantitate using some Leica confocal software. I turned the images into mean fluorescent intensity. So I gave the pixels a number, basically, the software did, and I was able to normalize for the background. When you start comparing 100 different patient samples, you got different background on it, everything's different. The tissues are all, everything, tissue different, it's handled differently, the surgeon had it out for 5 minutes, whatever. There's a lot of variation going on there. So it becomes a challenge to normalize these tissues. We were able to pull it off and publish it in Cancer Research in 2007. In this case, you know, we have the antigen expressed in a high amount, low amount. It turned into a statistical game, but it came out. It showed that the more of this antigen, the less likely the patient was to survive and over survival time. So the lower the antigen expression, the longer the patients lived in that. Okay, and that concludes in 42 minutes. That's not too bad. My little section of this grid thing. And so this is my contact information. If you have any questions or what, you'd like to come visit LSU School of Vet Med, give me a call or something, and we'll get you hooked up. And if there's any questions, I can answer them at this time. All right. So, okay, I'm going to pass it. Do you want me to just pass it over?
Any questions from Monroe? No?
Actually, this is Kui from LSUS. I do have a question. You didn't mention the word confocal, but you were saying with the Leica software, the Z stack, as I understand it, is it similar to confocal, or is it does it have the confocal capability?
Oh, okay.
Confocal fluorescence.
Yeah. We actually have a confocal at the School of Veterinary Medicine. These images and the microscopes I described were all wide field. They're not confocal, not laser driven, not scanner based. This was all just wide field, two-dimensional. But it's the Z-stack functions are similar. They're exactly the same as the confocal, except the confocal is using lasers and scanners to excite and capture, where in this case, the energy is a lot lower in this particular compared to a high energy laser system confocal. This is just a fluorescent UV lamp projecting across the image, across the tissue to exciting, and it's capturing it with the CCD camera, but it's recombining them in the same way that a confocal would with the Z-stack. Now, the big difference, the confocal lasers are designed to dig down into the tissue where these lamps won't. If you have a thick section and you try to illuminate in Z-stack form deep into the tissue section, most likely you're going to lose intensity just because the light won't travel down into the tissue very well. And then lasers, it does. It goes through the tissue really well. So that's the big difference between this type of Z-stack and with just the regular wide field, wide light and the confocal light. Does that answer your question?
Yeah.
Okay.
Pete, remember also we're looking to purchase a confocal, self-contained confocal to put into this core as well.
Yeah.
So,
Yeah, we're adding some more microscopes. We're looking at some confocals now, modernized confocal systems that are user-friendly and stuff. We hope they're user-friendly. At least that's what the salesman said, you know. So,
must be true then.
must be true.
Okay.
We have a question here at Louisiana Tech.
Okay.
In our lab, we study the Alzheimer's disease tissue, brain tissue, and we are beginning to exploring the amyloid plaque relationship to the modified protein. So what particular microscopy do you recommend here?
In brain tissue?
Brain tissue.
Yeah, you know, in this case, Dr. Alvarez is he's done a lot of research with brain tissue, and he's going to show you what the confocal will do with the brain. I've done brain tissue also. What I would do is I would start with the wide field microscopy, just regular fluorescence in the lab. Get your stains to work real nicely. You know, get your antigens, your antibodies to recognize your antigens with and your background down, and a nice fluorescent image that you can see in a 2D wide field fluorescent. And then once you've got your stains worked out really nicely, you could go to the confocal, and then your resolution will increase because you're able to get down into the tissue, like I said, with the lasers, and get a better image of what's happening deeper down into the tissue and that. But I would definitely start with the basic epi-Flourescence and to optimize all your parameters and stains, and then finally go to the confocal and to get a nice image. Because the brain slices, you could probably go up to 15, 20 microns, maybe even more than that, if you could get it to stick to the slide, you know, and you could really develop a really nice layer of cells, 3 or 4 cells in a layer in the brain. But you won't see that with epi-Flourescence. You'll have to go to the laser-driven systems for that, I think.
Well, yeah, thanks. Yeah, and I
Okay.
have a very, very, basic question here, but I don't understand. I would like to ask here. You mentioned about those white fluorescences. So basically, it's just white light and with particular filter for the excitation. Is that right?
I'm using that term for
Yeah.
what that is. Yeah, is the UV light source going through the optic, and that's what one of the ways to describe that is they call it wide light, and because it's giving you the across the spectrum intensity, excitation intensity, you know, across the spectrum from 300 nanometers to 700 nanometers or 8. Wide field go 800 nanometers? Yeah, to 800 nanometers. But it's a lot less intensity than, say,
Yes.
you know, a laser. The intensity is lower, the energy levels are lower because it's spread out.
Yeah.
But it's the entire spectrum, and you kind of filter out the piece that you want. That's why they call it wide field.
I
Yeah.
see. To avoid confusion, because I also heard about this mercury lamp and a halogen lamp. So they have the same capacity as this UV lamp, or what's the
The
difference?
halogen lamp is just generates bulbs like white, like in your house, you know. But it's a higher intensity, you know, wattage bulb for bright field imaging, bright field H&E slides and things like that. The mercury vapor lamp, and there's a bunch of different varieties of that UV light source that is the lamps themselves, different metals and things generate different spectrums, and some of them are you know, give you more intensity on the low end, the UV end. They go from the ultraviolet to, you know, the red, green, blue, violet to the infrared, the entire spectrum, and they have different, depends on how much you pay for them and which type you choose. Some of them might have a stronger intensity on the ultraviolet side, or they may lean to the infrared side. As far as, you know, if you were looking at an intensity wave, you know, it wouldn't be a straight line. It comes, you know, it would be different intensities at different spectral nanometer wavelengths. And it's all dependent on the product and the manufacturer, and then the life of the bulb. As the bulb gets older, sometimes it'll start to lean one way or the other, and give you different readings on that. But that's generally what we use with the epi-Flourescence here.
Thank you.
Okay.
Any more questions? All right, thanks for being here.